So this is the last section of our notes on cellular respiration, and in this section we'll be talking about the electron transport chain. One thing to keep in mind is that not every step of cell respiration is going to occur in every organism. So if you have an organism that is anaerobic, for example, well, we won't move much further past glycolysis and won't be able to continue on to the Krebs cycle. So just as a little review really quickly, just let's remind ourselves that glycolysis occurs in the cytosol, which is the same thing as the cytoplasm. In glycolysis, we start with a glucose molecule that is 6 carbons long, or 6 carbons big. And we start with just one of those molecules. In glycolysis, we actually convert the glucose into 2 pyruvate molecules, which is a 3-carbon molecule. As a product of glycolysis, we will produce 2 ATP, or 2 net ATP molecules, because we did have to use 2 ATP. And we also make 2 NADHs, which are the electron carriers that we need. Once we have produced our 2 pyruvate molecules, we'll be able to move on. If there is oxygen, then we'll move on to the Krebs cycle. If there is no oxygen, then we're going to do anaerobic respiration, and that's when we produce less ATP. However, it is faster, but we'll also make lactic acid or alcohol as a result. Okay? So once we have our 2 pyruvate molecules, then we're going to be able to move on to the Krebs cycle, which is also known as the citric acid cycle. Krebs or citric acid cycle. Remember, though, that our pyruvate does have to get changed, and it does actually have to move to or be converted into our our acetyl-CoA molecule. Okay, when we have our acetyl-CoA molecule, a reminder again that this is a 2-carbon molecule. Then it's going to go through the citric acid cycle, and we had 9 different steps that occurred here. In the citric acid cycle, we're going to produce a net of 4 carbon dioxide molecules, a net of 2 ATP molecules, 2 FADH2s, 6 NADHs, and 2 NADH2s actually come from the process of, how should I write this? The oxidation of pyruvate. Okay? Keep in mind that over here we had just 1 glucose molecule. We end up making 2 pyruvates, so that means we have 2 of our acetyl-CoA molecules, and so this Krebs cycle does happen twice. So at the end of this, we now have a total of 4 ATPs produced, 10 NADHs produced, and 2 FADH2s. So then we get to the electron transport chain. And a really quick overview of the electron transport chain is that, well, who are we working with? What are we working with? We're actually going to be working with our electron carriers. And with our electron carriers, we have 2 of them, NADH and FADH2. Both of these got oxidized and will lose electrons. This process, however, is going to occur in a transmembrane protein that is going to have a redox reaction occur as we pass the electrons along. So let me clarify a little. We have electron carriers that came from these previous steps. We're going to take the electron carriers, and the electron carriers in this form will actually get oxidized in this step. And that means we're going to lose electrons. This is going to happen in a transmembrane protein, so a protein that's along the membrane of the mitochondria or through the membrane of the mitochondria. And we're just going to keep passing along these electrons. And this is really what's key to this step in the electron transport chain. So what actually occurs, again, our electron carriers will transfer electrons to proteins, to different proteins, in the inner membrane of the mitochondria. Okay? When does this occur? This is going to be after glycolysis, after the oxidation of pyruvate, and after the Krebs cycle. Where does this occur? This is going to happen in our inner membrane of the mitochondria. And why does this occur? The electron transport chain, this passing of electrons through different proteins in the membrane, is going to occur because of the high energy of the electrons. There's actually 3 proteins that they're transferred to. And then to a final electron acceptor. In this process, our final electron acceptor is going to be O2, oxygen. And just keep in mind that oxygen is highly electronegative, okay? So we're going to pass along these high energy electrons. We're going to transfer it through to 3 different proteins. One of the reasons why that's possible is because of the high electronegativity of the oxygen molecule. And all of this is going to help, and actually the high energy electrons that we're passing along is going to help produce, or is used to build, a proton gradient. So let's use the figure of the inner membrane of the mitochondria to figure out what occurs here. So here's the structure of the mitochondrial membrane. Again, we're going to use these electrons to actually pump hydrogen ions to create a gradient that is known as an electrochemical gradient.
Jose Aguilar, please come to the front office immediately. Jose Aguilar. Teachers, if he's with you, please call the front office.
So for the structure of the membrane, we have an inner membrane. The inner membrane has just different compartments within it, and the compartments are going to allow for the creation of the hydrogen ion gradient. We're going to get our hydrogen ions, that proton, whenever the electron carrier loses an electron. So the electron carrier NADH or FADH2 loses an electron, and we gain a proton from that process. So here you have 3 different spaces, or 3 different locations of the mitochondria. You have the intermembrane space. Intermembrane space is going to be between the outer membrane and the inner membrane. So inter, or between the membrane. What happens is that within the mitochondria, we're actually going to have an electron from the NADH passing through some protein pumps. And right here I'm just writing in the word pumps. So this is an active transport process. As the electron passes through the first protein pump, It's going to be going to these different complexes, Q, another protein, cytochrome C protein, through another protein, and finally actually form water at the very end. So, what happens? We're really just taking the energy of an electron to change the shape of the proteins as it passes through. As it passes through, we're also going to have the movement of hydrogen ions. So as we move through, notice that our NADH2, we have this H plus here. We end up creating a gradient because we actually end up reducing the concentration of our hydrogen ions in the mitochondrial matrix and increasing, therefore, the pH. Remember that pH is the potential of the hydrogen ions. So when we have less hydrogen ions, we become more basic or have a higher pH. And on the other side, we notice that we're pumping up this hydrogen ions, so we increase the hydrogen ion concentration, which makes us decrease our pH, which means this area is actually becoming more acidic. So within the matrix, within the folds, we have a high pH or basic concentration, and outside between the intermembrane space, we're going to have this acidic concentration. All that's happening is each time the NADH carrying the electron moves through, we're changing the shape of the proton, or changing the shape of the protein, and actually causing these hydrogen ions that are within this area to be pumped out. So we're causing this area to become more acidic or have a higher concentration of the hydrogen ions. Then from there, we're going to continue to pass on electrons, and they're actually being passed to the oxygen that's waiting right here. Highly electronegative atom. So we're increasing electronegativity. Each of the following proteins are getting more and more electronegative, and the oxygen is the most electronegative molecule there. We end up forming some water here, and then at the very end, because we've increased the hydrogen ion concentration, now diffusion is going to occur. And diffusion is going to cause, and this is where we see passive transport, diffusion is going to cause the high concentration of hydrogen ions to move down through this protein called ATP synthase, or this enzyme. The movement of the hydrogen ions will cause a literal mechanical or physical shifting or turning of that protein. And when we turn that protein, we actually transfer this inorganic phosphate to the ADP molecule and produce ATP. So, what's happening there? We have, and notice that's oxidative phosphorylation. Oxygen's over here. We're phosphorylating or adding the oxygen here. So what we're doing is that this protein is permeable to hydrogen ions, causes a rotation, and there's actually a, we'll write in rotates due to the kinetic energy that's caused by the movement of that proton. So in the end, the kinetic energy is going to power the protein. So this is a physical energy, and I'm going to write E with a circle to represent energy, to form ATP, a chemical energy. And we're going to stop right there and talk about the final summary or the important points of electron transport chain. in class, but make sure you look back over this and understand what's happening with the concentration gradient of the hydrogen ions, and think about the processes of active transport that must occur from going from low concentration to higher concentration, and the process of passive transport when we go through diffusion. Okay, so this is what happens in our electron transport chain.